Okay, so last chapter we talked about how photosynthetic organisms converted the energy of sunlight to sugars. Um, and now we're going to talk about how all organisms, um, including photosynthetic organisms, use the potential energy in the sugar, in this case glucose, to extract high energy electrons out so that they can utilize that energy in the form of ATP for their own purposes. And so the way this works is that these sugars that are formed by photosynthetic organisms in all living organisms go through a process of glycolysis. And if you look at this, glyco, which stands for the sugar, glucose, lysis. Lysis means to split. So the process here, at least initially, is to split this sugar molecule up into two molecules and then extract the energy out of that. One thing to keep in mind here is that the sugar that we're going to talk about, glucose, is a six carbon sugar. And if there are two things to do, so it'll be split to two times three carbons, two three carbon species, is always kind of keep track of where your carbons are and keep track of where your electrons are, because the electrons are where the energy is. So in glycolysis, the sugar is split into a three carbon pyruvate. And then there is a choice that can be made here if oxygen is present. So aerobic is with oxygen, then you completely oxidize this pyruvate. You go down to as low as you can go, carbon dioxide and water, and then you form 32 ATP. In the absence of oxygen, so anaerobic means no oxygen, you don't completely oxidize this pyruvate. You generate one molecule of carbon dioxide, and then you have an organic molecule, which can either be lactate or ethanol, and two ATP are captured in that process. And so this process of cellular respiration uses a fuel and source of sugar and oxygen in order to form carbon dioxide, water, and ATP. Now, it is cellular respirations because the cell is respiring as we do. We, as organisms, breathe in oxygen and exhale carbon dioxide. The reason we do that as organisms is because every cell in our body utilizes oxygen, and we will see it utilizes it in the final as the final electron acceptor um, in the electron transport chain in oxidative phosphorylation, and we produce carbon dioxide as a waste product. So our body responds because that's what the cells are responding. And so we're going to talk about getting this energy from sugar, but we can also get it from protein, we can get it from fats, and in our biochemistry course, we go through how some of those processes occur. Now, if you look at this, we have as an input of cellular respiration, oxygen and sugar, and as an output, we have carbon dioxide, water, and ATP. Note that for photosynthesis, as an input, we have what? Carbon dioxide, uh, water, and then energy in the form of sunlight. And as an output, you have sugar plus O2, right? So here you can see that these processes of photosynthesis and cellular respiration are related, but obviously they aren't simply the reverse. If that were the case, we would produce sunlight as an energy source, and obviously we don't do that. So certainly they're related in a global sense, but it breaks down when you look at the details. So for us, this is why we have to eat. So we take in food because we are heterotrophic organisms. We break them down, we absorb the nutrients, and we use the energy from that in order to drive molecular processes. And then our cells do that as well. And so this is the um, chemical equation that we'll be dealing with here. Here we have the sugar. Remember, carbohydrates are hydrates of carbon. So for every carbon, you should have a water attached to it, or a molecular ratio of one carbon to two hydrogen, or to one oxygen. So you have your sugar in the presence of oxygen breaks down to carbon dioxide and to water and releases energy. The energy from the complete breakdown of glucose is 686 kilocalories per mole, but we will see that we only capture about a quarter of that energy in that process. And the way we trap it, how we capture this energy is in the form of ATP. So this is kind of like money or current or batteries. So this is energy that the cell can use. So we use the energy from the breakdown of glucose in order to drive this process of phosphorylating ADP. The D stands for di, which means two, and making a TP. The T stands for tri, is three, from ADP and inorganic phosphate. And this is the flow of the electrons in this system. So in cellular respiration, the final electron acceptor is oxygen, and that oxygen ends up in water. In the absence of oxygen, then in animals, the final electron acceptor is pyruvate. We actually go back to pyruvate. That ends up in lactic acid. That lactic acid, if there is an oxygen presence, gets pushed out into the blood, collects in the liver, where it reforms glucose. In organisms that undergo fermentation, like yeast, the final electron acceptor is acetaldehyde, and then it ends up becoming ethanol. And just kind of, this might be too much inside baseball, but pyruvate is three carbons, and you actually lose a carbon to form carbon dioxide here. So ethanol is a two carbon structure. So there's no going back here. In lactic acid, you can go back. In this case, you cannot. This is the process by which fermentation happens that makes our alcohol. It's also the process by which yeast produce carbon dioxide in bread baking and carbonation and all of those things. It is that they make this ethanol excreted out into the system. So we need to look at the transfer of electrons, and we went over this last chapter, so I'll just briefly reiterate it here, right? So a redox reaction is a oxidation reduction reaction, right? And you always have a transfer of electrons. Electrons don't leave one compound without going someplace else because electrons are high energy. And so we can transfer those electrons directly. Here you see this compound A has the electrons. It is reduced because it carries the electrons, and then it transfers those electrons to compound B, which is oxidized because it doesn't have the electrons, and compound B becomes reduced. In this process, compound A becomes oxidized. So you can transfer the electrons directly, or you can transfer them to an electron carrier like NADH. Now, in photosynthesis, we use the electron carrier NAD, NADPH. in order to carry those electrons. So similar basic construct there. So there, the electrons are passed in the form of hydrogen atoms to NAD plus to form NADH, and then that NADH can go someplace and pass them on. What we will see is that in glycolysis and pyruvate oxidation and the citric acid cycle, the electrons from the glucose are passed to NADH and then another electron carrier FADH2, and those will go into the mitochondria to deliver those electrons to the electron transport chain so that energy can be produced by the difference in protons across the inner membrane of the mitochondria. So that's where we're headed with all of this. And we've seen the structure of NADH before. Again, this is a modified nucleic acid. Oops, nucleic. And that's going to be used to pick up the high energy electrons from glycolysis and then transport those ultimately to the electron transport chain in the mitochondria. And so this is an overview of the whole system. We can break it down into really four steps. There's glycolysis, the splitting of sugar. This occurs in the cytoplasm and it doesn't need oxygen. From there, so you get a little bit of ATP from here, 2 ATP net in this process. And from there, the pyruvate molecules go into the mitochondria. And remember, we call the mitochondria the powerhouse. This is why, um, because it's the site of ATP production. And so the pyruvate will, one, go to form acetyl-CoA units, so it'll be broken down. This is where first carbon dioxide is produced. The carbon dioxide that we breathe out is first produced during the production of acetyl-CoA, and then those acetyl-CoA molecules feed into the citric acid cycle, or the Krebs cycle, where you produce the other, in this case, 4 molecules of CO2. You make a little bit of ATP, another 4 molecules of ATP. Oops, sorry, 2. Um, and then you reduce the electron carriers NADPH, sorry, NADH and FADH2. Those electron carriers then feed into the electron transport chain, and this looks similar to photophosphorylation and photosynthesis. The energy of the electrons are used to drive a concentration gradient of protons across the inner membrane of the mitochondria, and then the flow of those protons back, in this case into the matrix of the mitochondria, drives the production of ATP from ADP and inorganic phosphate via the enzyme ATP synthase. So it is a similar process. There are some differences. The thylakoid pumps protons out of the electron transport chain pumps protons out of the thylakoid um, in photosynthesis into the stroma. Here you're being pumped. Um, sorry, the electron transport chain pumps protons into the thylakoid from the stroma, and ATP synthase pushes them back out. Here, you're going from the matrix of the mitochondria, the interior of the mitochondria, into the inner membrane space of that mitochondria. And we'll see that in detail a little bit later on, probably in the next lecture. So glycolysis is used by every living organism. So this sugar splitting process is something that all organisms use to extract energy out of the sugar. So they could be prokaryotic, animals, photosynthetic organisms. Everybody uses this. And remember, this occurs without oxygen, so that any organism can get some of those. And the basis of how that happens is this. You have your molecule glucose. You activate it by investing 2 molecules of ATP. So you're putting in a little bit of money into the system. That is going to destabilize that glucose. Eventually, by adding that energy to it, you split it in half. And then in that splitting process, you extract 4 molecules of ATP and 2 molecules of NADH. So you put in 2, you get back 4. So you have a net of 2 molecules of ATP. And you form 2 molecules of pyruvate, and there's lots of energy still in that pyruvate. So in glycolysis, the net is an organic compound, ATP and NADH. Now you make 4 molecules of ATP, but since you invested 2 molecules, your net is 2 molecules of ATP. And this is a process. This is actually a 10-step process. In order to do this, each step is enzymatically driven. Um, and we are going to transfer these electrons to oxidative phosphorylation later on in the pathway. And so phosphorylation is simply the addition of this phosphate group onto something. Now, there are 2 ways that this can occur. It occur via phosphate substrate level phosphorylation. So that is when an enzyme directly puts in a phosphate to a molecule. And we'll see this a couple of times, particularly pyruvate kinase. And then you can have oxidative phosphorylation, in which the phosphate is added via a mechanism, some other mechanism. And so this is substrate level phosphorylation, and as we said here, this is the enzyme this is the enzyme pyruvate kinase that binds to ADP and one of the intermediates of the glycolytic pathway, phosphoenolpyruvate or PEP, and the phosphate is transferred from the PEP to the ADP to form ATP, and then pyruvate is comes out as a result of that. So 10 steps in glycolysis. The net result, 2 molecules of pyruvate, 2 molecules of ATP, net, and then 2 molecules of NADP, NADH. And this is the step I will say in biochemistry. We'll actually break down each of these steps and we'll look at we have to know all the intermediates and all of that stuff. I want to just kind of pull this up. This is actually a biochemistry slide just to highlight some kind of key things. 10 steps that do it. As we said before, the enzymes here, remember enzymes are named, they always end in ACE. right? And they're named after their activity and the substrate in which they act on. So phosphofructose kinase, it is a kinase, so it adds a phosphate. And what is it adding it to? It's adding it to a phosphorylated fructose molecule. And in fact, if you look over here, that's exactly what is going on. You're adding a phosphate to fructose 6 phosphate. Um, Here, I will say that anytime that you add an ATP, there's a significant change in delta G or the energetic levels. And so that's going to be a step that's highly regulated. So the step 3 here, phosphofructose kinase step, this is one where the cell is making a decision whether they're going to invest an additional molecule of ATP into this pathway. So they're only going to do that if the energy needs of the cell warrants it. And so this is a big decision point, and by inference, that means that this enzyme is going to be highly regulated. And you can imagine it will be positively regulated by indications that the cell is at work. So high levels of ADP, high levels of NAD plus, and it'll be negatively regulated by indications that the cell is at rest. So high levels of ATP, or high levels of NADP or NADH. Here, you split this 6 carbon structure. So let's see, 1, 2, 3, 4, 5, 6. You split the 6 carbon structures into 3 carbon structures, glyceraldehyde 3 phosphate, dihydroxyacetone phosphate. And then this next part is to pull off the energy from those from these molecules. And here there's a step that extracts the electrons in the form of NADH. There's a step that makes ATP, and then another step that makes ATP that allows for the energy to be pulled off from this destabilized program. And if you look at the delta G, we can see that you can kind of break this up into an energy investing pathway and energy harvesting pathway. You see, as you put in this ATP, you're raising the energy levels of this glucose molecule, starting from here, going up a little bit, and then as you begin to pull off that energy, you get dramatic drops in the delta G, all right? And then every time you pull off some energy, you see a relatively dramatic drop in the delta G. So this pyruvate molecule is at lower energy from the delta G, from the glucose that it came from, and it is because, and we've been able to capture some of that energy in the form of NADH, ATP, and and well, 2 molecules of ATP. Okay, so I'll stop there with this one, and then we'll do the citric acid cycle and oxidative phosphorylation in the next lecture.